You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I'm talking to Paco Calvo. We're going to be talking about the philosophy of plant neurobiology and the MINT lab, the Minimal Intelligence Lab. So Paco, thanks for coming. How are you doing today?
Hi, Richard. Well, thanks for having me.
Yeah. So you're studying plant intelligence, is that right?
I do, yeah. I know it sounds awkward, but that's what we do.
Well, I've spoken to some people that work with plants, and in many ways, plants seem unbelievably intelligent, especially in how they can create chemicals to defend themselves. And there's far more to plants, I think, than anyone thinks. So, but let me know your interpretation. What fascinates you about plants, and what got you into working with them?
Well, yeah, I guess that the starting point is definitely that we suspect that plants are smart, right? They've got to be intelligent. Otherwise, they wouldn't be here, right? So in a sense, they've got to be intelligent to have passed their genes, right? But I guess that we don't quite understand what we mean by that. So what do we mean by intelligence, or what do we mean to say when we say that plants are intelligent, right? So, for example, this thing about the chemicals and these things, that could be interpreted as an adaptation, right? You might just say, yeah, sure. I mean, they behave adaptively, but that might not deserve the label intelligence, right? So I guess we need to do some work, both theoretical and empirical, to really grasp what it means for an organism, for a living system to be intelligent. So that's what we're after.
Okay. So what are some examples of plants that really impressed you, that made you feel like, wow, this has to be a lot more here than we're seeing initially?
Yeah. Well, I guess generally speaking, I mean, any pattern of behavior that is anticipatory, so is, in a sense, they are predicting future outcomes, and is goal-directed, and is flexible enough so that it's not hardwired, like, responding to environmental sources of stimulation on a one-to-one basis, right? So like the cartoon picture of plant behavior is like, they sense gravity, they grow downwards the root, they sense light, so they behave phototropically. That's the cartoon version. The truth is that things are a little bit more complicated. So they have to integrate all this information coming from all these channels and somehow provide a response that is globally adaptive, right? So the examples in particular, that's a thorny issue, because for one thing, we miss most of what plants do. And that's our real hurdle. To be able to observe this sophisticated behavior that they are able to exhibit. And we simply miss it because of the scale of observation, their time scale, our time scale as observed. Here, for example, at the Minimal Intelligence Lab, what we do is we time-lapse plant behavior. When we say plant behavior, we mean their patterns of growth and development. So we have a plant, we time-lapse it with a camera. So we take a picture like every minute, every 5 minutes, and then we assemble a footage after a few days. And then when you do that, So when you slow down your scale of observation to the behavior, to the time scale of their behavior, then you are able to start unearthing some patterns which are really interesting. In particular, in our case, we are especially interested in climbing beans or climbing plants in general. In particular, the climbing bean is one of our models. And here we try to understand how is it that they control if they happen to do, because that's another issue, whether it's actually the case. So, but the working hypothesis is that they might be controlling endogenously the approaching maneuver. So the way they target a support, a pole, a host, somewhere, some support to climb onto and trying around like good climbers do, right? And we try to understand how is it possible for them to do it without a brain, because we've got to remind ourselves that they don't have a nervous system. So how are they able to do it? So that's one of the challenges, and the climbing bean in this case, or vines in general, are a challenging example of non-neural intelligence.
So what have you observed? I guess you set up a whole bunch of plants or climbing beans and a whole bunch of poles, and then you've watched using time lapse how their stalk would approach the pole and wrap around it. And looked for behaviors that tell you if it's sensing the pole and deciding whether to wrap around it at a certain angle, and or just blundering around and feeling the pole randomly. I mean, like what have you observed specifically when you've looked at this kind of stuff?
Well, here, we do two things actually. Technique I was telling you about. On top of that, we are also registering the electrical activity going on throughout the plant body with electrodes. We insert electrodes throughout the stem. So by combining these two techniques of observation, we are able to gather more data and try to find correlations in between the overt behavior that we are able to observe through time-lapse footage and the underlying electrical activity. Now, we've got to be a little bit cautious here, right? For one thing, right now, I would say I'm more interested in, rather than in providing, like a set in stone answer, which we don't have yet, and hopefully we won't have for a long time. And by this, I mean that in science, we should be more interested in the type of new questions we are able to put on the table, rather than the, mainstream or orthodox answers we are able to provide. So at this stage, I'm even more interested in trying to unearth new patterns of behavior, things that we weren't expecting them to do. And in that sense, we are, we've been time-lapsing round the clock for the last couple of years. And we want to hold it back and be careful enough, not to overinterpret the data. We want to be careful, very careful, and avoid, anthropomorphic biases, because for one thing, the way, in a sense, when you are watching time-lapse footage, you can't help but, project your anthropocentric biases and preconceptions onto the footage you're watching. So we need to be very careful, and then we need to, to sit back and relax and think of themselves, of the subject models, and not of ourselves as observers. So for example, now we digitize all the time lapses frame by frame, and so that we can track down the actual trajectories and then see the patterns of acceleration and deceleration. So take, for example, if you see the vine approaching the pole, you might expect them to accelerate or to decelerate gently, as think of a bird landing on a perch. So to speak, they might be doing the same, but we don't know. And this is a complex issue. So at this point, we are trying to collect as much data as possible before we are able to, to make a pronounced cautious.
Well, if you have years of time-lapse data already, you're not analyzing it, or are you analyzing it? Like, what have you seen so far? What are some interesting things you've seen?
Well, we suspect that there is definitely a form of endogenous control which means simply bump onto the host or the pole, so they don't simply, circumnutate. So the pattern of circumnutation, this is the type of movement all plants exhibit. Actually, any plant organ, so could it be flowers, leaves, stems, tendrils, roots, shoots. So any part of a plant grows by performing this motion of revolution, right? The pattern of circumnutation, it revolves around as cells elongate differently, right? So one
You mean
cells.
a tendril will spiral outwards in a direction? Is that what you mean? Instead of
yeah. So
growing straight, it spirals out.
Yeah, that's it. So they revolve around, and in a sense, that's the regular pattern, right? So in that sense, as remember we were talking before about behavior that is merely adaptive. So you might say, hey, no big deal about it, right? So vines certainly are in the business of climbing around hosts. So definitely those that are good at that are the ones that pass their genes, right? So in a sense, we want to put some pressure on them, even if artificially, of course, in an experimental setting, but see how they are able to cope with uncertainty, with contingencies. Put it this way. We were saying that plant behavior, on top of being adaptive, is got to be anticipatory or predictive or goal-directed, right? So another way to put it is that precisely because plants operate on a slower time scale, they, in a sense, they can't afford to miss, right? So imagine you human or an animal, you might say, hey, I can perform this pattern of movement. I can try to grasp the mug or just the pole or whatever with my hand. So I stretch out and then try to reach out. And then if I miss it, I can try again, no? In their case, because they don't move by locomoting towards the target, but rather by growing towards it, right? They might not be able to have a second chance the way animals do, right? So because they are sessile and they are rooted, and the way they deal with their environment is different from ours, they might not be able many times to have a second chance. So in that sense, anticipatory behavior is a must. So they can't afford to miss. And that's why they have to be somehow endowed with smart mechanisms or ways of dealing with those goal-oriented needs that they have, right? So in a sense, what we see,
Okay.
yeah, sure, go on.
Have you done an experiment where a plant will grow towards a pole and then you move the pole and see how the plant reacts?
Yeah,
that'd be a simple test.
Yeah, that's on the agenda. I see what you mean by being simple, but believe me, it's anything but simple. That's definitely on our list. We have many experimental variations we are trying. We don't, as I insist, we don't want to be careful because there are so many things that, so many alternative explanations. And it's so easy to fall prey of confirmation biases that we don't want to fall into those biases. So actually, you know what happened to me once? I had this vine. Okay, I set the time lapse, the settings. Everything was ready to go. It was a Friday. And I said, well, perfect. I mean, this is, by the way, this is not like Darwin, right? So Darwin would make observations to the naked eye, right? So he would have to be real patient and be there all day long, marking on the glass. He was using a glass plate technique, like marking, aligning the tendril or the tip he was interested in with the background point. He would align them and then mark it on a glass in front of the plant. And then he would need to do that on a regular basis. So he had to be there, right? So in our case, time lapsing is much easier. On a Friday, I just prepare the settings, a picture every minute, off we go. And then I can go for the weekend and relax. And then when I come back on a Monday, I can check it out what is done for the last 48 or 72 hours, right? So this is what happened. Once I got back to the lab one Monday morning, and there was, the vine was, here was the pole. The vine was lying on 180 degrees from the pole. So it had just gone the opposite direction, right? Complete opposite direction. And I realized how lucky I had been in this case. You know why? Because imagine that the vine happened. Imagine it had happened to land onto the pole. So the way it was, but the other 180 degrees, right onto the pole. I might have thought, hey, look at that. It went straight for the pole. But now, because it had gone right the other direction, I was able to understand what was going on. In this case, I found out that the matter it could be explained simply on it was a matter of physical constraints. It seemed that the soil in the pot had been soaked far too much when we watered it, and it had lost stability. So it didn't have any support, and it just went all the way down because of the water, because of the pot. The soil was so soaked. So, but imagine, I said I was lucky because this way, that allowed me to control more carefully about the way to water the pots and everything, how to hold the stems without interfering with their natural patterns of growth. But if in this case, the plant had gone all the way towards the pole by mere coincidence, I might have overinterpreted the time lapse and say, hey, look at that. It went straight for it, right? So that's a very stupid example, of course. That's something that if you have proper controls, you are able to discard, right? If you time-lapse systematically. But the amount of parameters that you have to deal with, it's enormous. So round the clock, and there is always some other alternative explanation that you have to take care of. So that means more pilots, more controls. So the case which you were telling me about the shifting the target, we are definitely working on that as well. So all combinations, including different genotypes, because not all climbing beans behave the same way. So different genotypes, different photomorphology affects as well. So the way plants shape up their plant body out of the light regime, so how light affects the way they grow. So because they are reaching the pole by growing taller, then there will be a displacement of the center of mass, and that's going to affect the way they change this initial pattern of circumnutation into the elliptical trajectory they would follow when they are targeting the pole. As you move the pole, it also bears not just the shifting of the pole, but how you are the temporal scale in which you are moving the pole, because it's got to be within the reach. So that's to say, you might think they are missing the pole when you move it, but it's got to be within the reach. So in terms of their patterns of growth. So, and that in turn has to do with the temperature, the circadian rhythms, because of the way they are regulating.
What do you mean? What do you mean within their reach?
Yeah, well, imagine. Yeah, if you are moving the target progressively away from the plant to see how the plant keeps trying to target it, right? The plant might be perceiving the support at a distance, which that's another vexing issue. So what's sensory modality is involved, right, in their perception? But anyway, leaving that aside for the moment, they might be perceiving the pole and yet not being able to reach it because the rate of growth is not within the scale of the distance you are moving the pole. So you might be moving the pole at a pace which is out of their rate of growth possibilities, right? So they grow as much as they can within their natural conditions. If you put it too far, they might not make it, not because they are not trying to, but because it's without their. So you've got to try to make it as the setting as natural as possible. This, in a sense, this also takes us to there is always, as you know, a tension in between artificial experimental lab conditions and natural settings, right? So there are two issues here: is these conditions how fair they are, right? You might be testing plant behavior in a setting which has anything to do with their natural conditions, and then you might be creating an artifact or making them, or actually triggering behaviors that wouldn't happen, wouldn't take place otherwise. Or you might be considering, when you time lapse in the open, in the wild, you might be discovering patterns of behavior that simply don't take place in the lab because it's a less rich environment, right? So there's
well,
far
why
too
not
many
start
things.
with the Why not start with the initial conditions? Like you mentioned, a good hint you got was that one plant you had watered the soil so much that the soil wasn't structurally stable enough
Yeah.
for it to reach out to a pole.
Yeah.
So maybe start with conditions there. Look at plants that have different depths of soil, different moisture amounts in the soil, different types of soil. The same plant, and see how that affects reaching out for a pole in the same place, the same distance, that kind of stuff.
Oh yeah, sure. I mean, the number of parameters and things you have to bear in mind is enormous. So it's this thing, and it's so many others. So we are trying to to take a look as many different conditions and settings as possible. So in that sense, you were insisting like, well, but what have you been able to see so far? And I've been telling you that we want to be careful. And the reason is that nowadays there is a risk. Especially in these fields where there is, in a sense, some resistance from mainstream science, from orthodoxy, and in a sense you have like an extra burden of proof, right? And to make a strong claim that plants are intelligent, you need to have real robust data and a very good case. And nowadays we live in a world of academically speaking, this is a hectic world of, the infamous publish or perish, right? Things like that. And I don't want to play that game. I want to slow down and take it easy and think enough about it, because my interest is understanding what plants are up to, not trying to show that one position is correct and another incorrect. So put it this way, I don't want to show that plants are able, or in this case vines are able to control their approaching maneuver, as we said before. Not that, but even something which is even more modest. I want to first find out whether they do it, and in case they do it, then to find out how they do it. So to do that, you cannot be using the opponent's or alternative explanations, alternative hypotheses as strawman alternatives. So you want to give your rival, your opponent, their best chance. You want to have a very strong alternative explanation, so that if you do happen to provide a good explanation, or to have good data that backs up your hypothesis, then it's also robust enough. So we want to be, I insist, we want to be cautious and try to find out about it. It's all we want to do. So here is not, there is a thing, for example, now, for example, the Templeton Foundation is doing something which is very good, very interesting, and it's called this, adversarial projects, where you team up with, not with the sympathizers of your working hypothesis, not the guys that would a priori be willing to buy your theory, but actually you team up with the guys who would be willing to defend another view, so that you can have different predictions and the possibility of making experiments that will tell in between them. So that's the way to go, because it's not my hypothesis against yours, it's all of us together teaming up against ignorance, right? So that's the way I would like to see how we should be doing science.
Well, it appears as the neo-Darwinists have a stranglehold on thought, so they want to say everything is random mutation and things like that. And if you're of that thought, then anything you do, they're going to try to pass it off as that. So it's, it appears to be a huge problem. Doesn't appear to be there's much evidence for many more mechanisms of evolution than that. But I don't know if you run into that at all. But I've spoken to many scientists that seem to unfortunately live in fear of that interpretation, that they'll be made fun of if they don't ascribe to it.
Yeah, I don't know. Yeah, I don't know. Yeah, it's a difficult issue, but nowadays again, Darwinian. In a sense, we have to go back to this to the botany of the 19th century, And by this, I mean, to Darwin had a clear insight, which is that botany, in a sense, wasn't subordinate to zoology, right? So that's, in a sense, that's why I think it's interesting to look into plants, not only because we want to, from a basic science point of view, we simply want to understand what they are up to, in the very same sense that we want to understand scientifically any phenomenon in the universe, right? But in a sense, because by thinking differently about plant life, we might be able to throw some more light on our own intelligence, human intelligence, and rethink those Darwinian terms that we started from, right? So think nowadays with all the revolutions in epigenetics and many other vexing issues. But, in a sense, we, not even plants, but more generally, the whole tree of life, if you put it in terms of the tree of life itself, then, a very complex pattern, which in a sense, paradoxically, is a very simple pattern, starts to emerge, that basically, and this is what was also underneath Darwin's position, is we are not that special, right? So we are just a tiny end of a branch in the tree of life, and something unites us all, right? Take the very notion of sentience or consciousness. It didn't, pop up from scratch in human or other, higher animals, but somehow it's got to be understood in terms of the very tree of life itself. So, if you allow me to put it in this way, I would say that in a sense, where there is life, there is already mind, there is already mentality, right? The only problem we have in understanding this motto is that we cannot help but thinking anthropocentrically. And that's the fight we've been fighting this battle for centuries, like, reminding ourselves that we are not the center of the universe. We know the story. I mean, but, it seems that the Ptolemaeo-Copernican revolution, the shifting towards, putting ourselves in this tiny corner of the universe is happening nowadays in the neurosciences, in the cognitive sciences, in the brain sciences in general, to try to remind ourselves that we are not that special, right? Maybe plants allows us to understanding, or studying plants will allow us to see it under a different light.
Well, look, all this being said, anthropomorphic or not, what other behaviors have you seen in the footage? You get a lot of footage there, I mean, you told me one, which was a good hint, but what else have you seen? Whether you can explain it or not, that piques your curiosity.
Well, we work with three plant models. One is the climbing bean I told you about. Then we also have a Mimosa Pudica, right? This is, well, of course, listeners will know about it, right? The guy that falls when you touch it. That's for Mimosa. You don't even need to time lapse. You can see it to the naked eye. And then we also work on roots of maize, right? Those two other projects, now we started like a project this last January with these three models. So it's those two others we are at the very early stages, and we wouldn't be able to report any data yet. We will soon be able to start doing something, but at least in the case of the climbing beam, some other behaviors which are interesting. One actually that I'm really, really curious about is that some beans. Well, these beans, they mostly all revolve anti-clockwise, right? So they go anti-clockwise, and then you can see how they are actually changing, or the circular pattern of circumnutation changes progressively into some elliptical shape as they are targeting and approaching the pole. In effect, when you move the pole around, you see there is some reports of this during the 20th century, last century, all the way. There is some very good research by French plant physiologists, which, by the way, this was a literature that, because it was in French and never got translated, it didn't go into mainstream community. So many people are not aware of this literature. But anyway, there is some very good literature from the 60s in France, and here you already have the grasp the understanding that plants were tracking the target as you were moving it around, right? But here, what we've been able, on top of this, what we've been able to observe is that these plants perform an incredibly rapid clockwise direction. So if they were revolving anti-clockwise, when they touch the support, sometimes rather than making miss it, they slip through. So they go, I touch it, but then they have to wait and make a whole new circle of revolution before they are able to touch it again, right? And try to hold on to it. Sometimes they are able to move backwards, so that would be clockwise, at an incredible speed, and that's something I had never seen before. So I have some footage.
Oh, they reverse their their spiral back out.
That's it. But not just that. But what really grabbed my attention was the speed, because usually think that these patterns of this, so to complete a whole revolution. This is temperature dependent mostly, but I would say that the fastest lap, it takes them like an hour and 10 minutes, an hour and 15 minutes.
You've seen some of them reverse back out much quicker than you anticipated. So what's the relative speeds of spiraling in versus spiraling away that you've seen?
Yeah, so that's what really, what really caught my eye was that as they were reversing this pattern, when they do perform the natural, the anti-clockwise movement, they might, it might take them like an hour and 10, an hour and 15 minutes to complete a whole circle of revolution. Now, when they reverse, after having touched the pole, when they reverse the direction of their circumnutation, they are able to complete half of the trajectory, which would have taken them like 20 or 30 minutes. They are able to do it in 1 or 2 minutes. So that's unbelievable. I've watched this for a few times. I joke about it. I say this is the Usain Bolt, the race.
Right.
So this is the Usain Bolt, but still, I go back again to being cautious. I don't want to overinterpret it. It might be, there could be many possible explanations of this, but this is certainly intriguing. So we want to keep time-lapsing and see what we find out.
Well, for instance, there you said you want to be cautious. What could be several interpretations of reversing quickly?
Say again?
What are some possible interpretations? You said you want to be careful about how you're interpreting this. So what are some possible interpretations that you think would be,
well, for one thing, sure. For one thing, as the tendril touches the pole, sometimes you can see how there's some tension building up, right? It's just barely touching with the very tip of the tendril, is very touching the pole, and it keeps putting pressure to keep moving anti-clockwise, and then you see the bending zone. The bending zone is curving, and is building up some tension. So it might be simply be a physical constraint that is like a spring-like. So it would spring back out of to release the tension. So it might be just a physical explanation of the phenomenon. That's why we want to be extra careful.
Okay. Yeah, I hear. So you think it may just be physically all of a sudden it took the plants 20 minutes to do one action, but now for some reason it takes 1 minute, but that's just something physically going on that's allowing it to do it. There's no intent there. It's just a reflex somehow.
Yeah. That's an alternative explanation, and we shouldn't ignore it. as I was insisting, we should not make of our, of the contender's position a straw man position. So let's give them their best chance to counterattack, and then we might be able to design other experiments and make more accurate observations and try to discard them, or not to discard them, but to see what's actually going on there. We don't know yet.
Okay, well, very good. So what's the best way for people to find out more about your work and maybe to get in contact with the lab or ask questions?
Sure.
Should they email? Should they go to a website? What's the best way for people to follow up? Find out more.
Yeah. Well, of course. I mean, well, I don't know if this is good or bad, but I'm usually on my email, right? People are welcome to drop me a line, and I usually will reply. I try to respond to all the emails. If there are too many, it will take some time for me. So please be patient. But email, I would say, the best way to reach out. But of course, they can also check out my website and which is Minimal Intelligence Lab. Here at the University of Murcia. So they can Google me up and see what we are up to.
Okay. Well, very good. Well, Paco, I appreciate you coming on the call. It's been interesting. Thank you for being here.
Well, thanks, Richard. It was a pleasure.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.